Okay, we're going to continue to think about phylogenetic trees, and now we're going to think about how we would actually go about making one. So, to do this, we're going to use a hypothetical example here. Consider these three birds. We're going to consider these three birds to potentially be related to each other, but we might start to notice that they have some differences in their phenotypic characteristics. Species A has a crest, a hooked bill, and has webbed feet. All right, so all three of those characteristics. Species B does not have the crest, but does have a hooked bill and webbed feet. And species C has a crest, but not the other two characteristics. So, we want to think about what an evolutionary history between these three species might look like. And we could start to imagine drawing a phylogenetic tree to represent these relationships. So let's imagine for our purposes that these three species came from an ancestor that had none of those three characteristics. No crest, no hooked bill, no webbed feet. One possibility, right? We might recognize this as being a polytomy. We have all three lineages splitting off from the same common ancestor. And so that means in order for species A to have those three characteristics, they all have to evolve on species A's lineages. Species B has to have the hooked bill and the webbed feet evolve, and species C has to have the crest evolved. All right, so I'll remind you that this is a hypothesis. This hypothesis would represent what we see in our final three species. And so this is an appropriate phylogenetic hypothesis about the way these three species could be related. Another possibility could be something like this, where we have our original split from our common ancestor. Species C diverges off by itself. Species A and B diverge from each other later, so they share a later common ancestor. And in this example, we have the hooked bill and the webbed feet evolving before that split for A and B, and then the crest evolving later. Another appropriate hypothesis has all the characteristics we need. Third possibility, maybe instead of A and B being sister taxa, A and C are sister taxa. So in this case, the crest evolves before A and C split from each other, and then the hooked bill and the webbed feet, well, those things evolve independently in two different lineages. So as we look at these three, all three of these have the appropriate characteristics. We have the three species that we end up with. And so all of these are appropriate hypotheses to explain the evolutionary history of this group. But we now then need to make some decisions about which one of these is most likely. And so the first thing we're going to do is let's add up how much evolution is taking place. By which I mean, how many times do new characteristics have to evolve in these lineages? So if we look at the lineage on the left here, we have the evolution of webbed feet, hooked bill, and crest. Three different evolutionary events leading to species A. Species B, we have the webbed feet and the hooked bill. That's two more. Species C, have a crest evolve. So that's 3, 4, 5. That's 6 different new things had to originate in that phylogeny. If we look at the middle one, we've only got 1, 2, 3. We've got 4 new evolutionary events. And then at the far right, we got 1, 2, 3, 4, 5. We've got 6 again. Oh no, I miscounted. We have 5 again. We have 5 over on the far right. All right. So that means that these three phylogenies differ in how many evolutionary events need to have taken place. Well, in order to make the decision then about which of these is most likely, we're going to use a concept called parsimony. And the basic idea of parsimony is that it says that the simplest idea is most likely to be correct. That whatever idea requires the least amount of assumptions or the least amount of new things, that simplest is best. And so if we look at our three phylogenies here, we see, well, the one in the middle is definitely simplest because it only requires 4 new things to evolve, whereas the other require more. So, barring any other evidence, right, barring any fossils, for example, that could help point us the correct way, we would say that the middle phylogeny there is the most likely. It requires the least amount of stuff to happen. Only 4 new things have to evolve, as opposed to 5 or 6 for the other 2 phylogenies. We are going to use that concept of parsimony to decide, amongst different phylogenies, to try to decide which one is most likely. Now, let me just mention that what we've done here, we were able to come up with these 3. There's a process we kind of call maximum likelihood, which is this really complex mathematical calculations that have to be done by a computer. That is going to be used when we start talking about larger numbers of species in a phylogeny. For example, let's imagine that we have 50 species in our phylogeny. All right, maybe you're studying, I don't know, a family of birds or a single genus of beetles, maybe. And there's 50 different species, and you want to figure out, all right, how are these 50 species related to each other? And so you start drawing those branching diagrams, those phylogenetic trees that we were just looking at. Well, as you start drawing them, pretty quickly you're going to realize that there's a lot. That there's lots and lots and lots of ways that 50 species could be related to each other. Right? That species 1 could be a sister taxon to species 2, or it could be a sister taxon to species 3, or 4, or 5, or 6, or 7, or 8, or 9, or 10. You see what I'm saying here? In fact, if we have 50 species, there are 3 times 10 to the 76 different trees that could be drawn from that. I just want to make this clear. That's a lot. So many, in fact, that there's no way that you could ever draw them all out, right? You would die before you had all of them drawn, let alone then going through and figuring out which one is the most parsimonious. So maximum likelihood is basically this computer model that goes through and does those calculations for you, because obviously it's impossible to a single person ever to do all that. Generally, if we're talking about a group of 50 species or more, you're going to end up with more than one tree that is equally parsimonious. And so the results of the model might say, well, this tree and then this tree are equally parsimonious, and this one only has 1 change difference or something, right? And so that'll give you sort of the options of the evolutionary history of that group. The more information that goes in, the better confidence you have, but it's a huge amount of work. And that's why we have computer programs to do it for us. All right, so let's imagine that we've gone through and a phylogenetic tree has been created. And maybe it's this phylogenetic tree here with a bunch of animals we're familiar with. We can start looking at that tree and start interpreting what that tree tells us. One of the things that we're going to see on this tree is characteristics known as plesiomorphies. A plesiomorphy is an ancestral characteristic that gives us a broad idea of how these species are related. It kind of is the characteristic that tells us about the big picture of a species. So in this example, if we take a look at our current phylogeny, and we see what kind of species we have here, some plesiomorphic characteristics, some ancestral characteristics for the group, something that would have been found in this common ancestor here. Could be something like, let's see, bilateral symmetry, right? Having a left and a right side. Eyes. All of these species would have had eyes in the common ancestor. Being made of cells, right? That's an ancestral characteristic found in the common ancestor, passed down to all of the descendants, right? So plesiomorphic characteristic is kind of that big picture characteristic being the common ancestor of the whole group. There are also going to be some characteristics then that we're going to call synapomorphic. And we're going to say that this is a characteristic that would not be found in the common ancestor, but is derived. So it's from later on, and it helps us to figure out who is related to each other within that broad giant group. So our example here, maybe we were talking about the mammals. So down here in the bottom right, we have this phylogeny, this specific part of it showing the mammals. We say, what's some synapomorphies for mammals? We can say, well, you know, hair, for example. Hair would not be found in the common ancestor of this whole phylogeny, but it is derived later on, found only in the mammals, tells us the mammals are related to each other. Mammary glands, the characteristic for which the group is named, would be found just in the mammals. A dentary squamosal articulation of the jaw joined with the head is found only in mammals. Synapomorphies help us to figure out who's related to whom within that broader group. Now, I just want to point out that plesiomorphy and synapomorphy are relative terms. We have to be looking at a specific phylogeny to figure out what they refer to. If we were looking at a phylogeny of just the mammals, then hair would become a plesiomorphy, right? If we just cut the mammals out, then hair would be an ancestral characteristic, and so would not tell us about how any of the mammals were related to each other, right? So it all depends on the specific phylogeny we're looking at the moment. Okay, so we've talked about then the way that these species diverge from each other and are representing that through this drawing known as a phylogenetic tree. Let me just mention that, like a lot of things in biology, there are some complications. For example, if we normally think about this kind of nice dichotomous branching, right, where a single species diverges into two lineages, and then each of those can diverge, right, that at each of these spots there's a divergence into two. And certainly a lot of evolution probably happens that way. But there's this other thing that can happen known as horizontal gene transfer. And to understand what I mean by that, first let me just mention what I would mean by vertical gene transfer. So in vertical gene transfer, you have parents pass on genes to their offspring, right? Vertical, down through the lineage, that a parent provides genetic information to the offspring. Well, that would mean that horizontal gene transfer, you would have an individual providing genetic information to another individual that is not its offspring. And so DNA going from one individual to another, but those two not necessarily being related, maybe not even being the same species, but you still have transfer of genes. In fact, there's actually, you might have seen a movie that's kind of about this. I don't know if you've seen the Sisterhood of the Traveling Pants. That is a good example of horizontal gene transfer, where one person has those genes and they transfer it to somebody else who's not their offspring, who can then use those genes. Sisterhood of the Traveling Pants is basically about horizontal gene transfer, which takes our ideas about that nice dichotomous branching and changes them to be a little messier, where we have genetic information being transferred maybe all the way across the phylogeny to not closely related organisms. We as humans, we don't do this right now. I mean, I've tried it. I've tried to go out and take leaves and rub on my arms so that I can become photosynthetic. So far, nothing. But in some organisms, this is something that's relatively common and really changes the way we think about relationships, because we don't necessarily just have that nice dichotomous branching of parents passing genes to offspring, but we can also have horizontal gene transfer, which, as we'll see, we think has been pretty important in the history of life on Earth. In fact, some people think that if we look at those earliest life forms, that maybe instead of having like a single ancestor that diverged and diverged, that maybe there was this kind of like pool of prokaryotic organisms that were constantly passing genes to each other. And And then eventually from that, our three major lineages kind of diverge out, but they don't diverge from a nice single common ancestor, but instead from this related group of prokaryotes that are kind of just randomly trading genes with each other. So we'll see more about that later in the course, but just keep in mind, as much as we like to think about phylogenies as that nice dichotomous branching, they aren't always that neat and easy. So I want to think now about the origin of life, right? Because really, if you wanted to, you can divide the history of life into two major steps. One, life begins, right? You have to have the start of life, and then after that, you have those large-scale evolutionary changes. But none of those are possible, right, without life starting. So really, the major steps are life begins, and then evolutionary change happens to result in that diversification that produces all the species we now have. And you don't have to write these down or remember these, but these are just some examples of kind of those big scale things that have been really important in the history of life. But none of those things can happen without the beginning of life, without the origin of life. And so what I want to talk about then is that first step, that origin of life step, that step that makes macroevolutionary changes possible, and what we think about how the origin of life might have happened. Now, I'll point out again, this is not really an evolutionary question. You can only really have evolution once you have living organisms. This is a question of how do you take non-living stuff and make it living. We're going to think about how this happened, and we've sort of got some ideas about the steps that must have occurred. Now, let me just mention going in here, we do not have great evidence for a lot of this. A lot of this is just sort of, we know some ideas about what must have happened, but we, in most cases, don't know how it happened. And you'll see that as we go through. Okay, so in order to start having living organisms, a possible first step is small organic molecules have to be produced abiotically. Abiotically meaning without life. So, for example, hopefully you recognize there is an amino acid. The second step then is those small molecules need to be put together into larger molecules we call macromolecules, a bunch of small molecules attached together. Those small molecules then those large macromolecules can end up in what we would call protocells. And a protocell is basically going to be a membrane around some of those macromolecules. And then finally, an important last step to have actual life is you have to have molecules that can replicate so that you can pass on information from one generation to the next. So once you have all four of these steps, then you can have a living organism. So we're going to talk about each of these steps and what we know about how it might have happened. Okay, so step one. We need those small organic molecules to be produced abiotically. Alright, so we think the Earth was formed around 4.6 billion years ago. Probably through a process of ice and rock slamming together. You end up with this solid structure that is a planet. Around 3.9 billion years ago, things had probably cooled off, and the Earth was just kind of hanging out. And at this point, the atmosphere was quite a bit different than what we have now. Here were the components of this early atmosphere. Alright, so water vapor, nitrogen gas, carbon dioxide, those are all things that we have quite a bit of. Methane, we have some of now. H2S, this is hydrogen sulfide gas, smells like rotten eggs. If you ever get a time machine, I would not recommend coming back to 3.9 billion years ago because everything would smell like rotten eggs. And then also hydrogen gas. You might also notice something missing there, something that we have a lot of today, that is to say, oxygen. So no oxygen gas. So the atmosphere is different then than it is now. There would have been a couple of possibilities of energy that could have been useful in creating these small organic molecules. Specifically, lightning and sunlight would have been potential useful energy sources that could have helped to form those early small organic molecules. Well, so because we sort of have some idea about what the Earth's atmosphere was like and energy sources that would have been available, some scientists, the most famous of which being Stanley Miller, decided to try this out to see if small organic molecules, things like amino acids, could potentially be formed under those conditions. So this is a picture of someone clearly doing science. And this is a replication of what the early Earth's atmosphere would have been like, right? So those gases we talked about would be here. There's the early sea, right? It's probably got a Bunsen burner, so keeping this warm. And so water, those other gases, and then you can see he's holding this electrode here, putting electricity through, so a spark would jump across, which would provide then sort of a simulation of the lightning maybe going through the atmosphere. And then every once in a while, Stanley Miller would take a sample of the water here and see what he found. And what they find, well, they did. They found small organic molecules. They found the sorts of things that could potentially be built together to make the large molecules that are important for life. That abiotically, things like amino acids were formed in this non-living environment. Now, I will mention that since Stanley Miller's experiments, there's been some changes in our understanding of what the chemistry was like in that early atmosphere. We think that it was a little different than what Stanley Miller used, and the new version, the better understanding version that we have now, the abiotic synthesis of small organic molecules has not been able to be replicated. So, I mean, the big picture that abiotically small organic molecules can be replicated, that was shown by Stanley Miller, but we don't know exactly how it happened because the chemistry is different than what Stanley Miller would have used. So, possible, but we don't exactly understand how it happened. Some people have argued that maybe there's another option. There are these rocks that occasionally fall from the sky that are known as carbonaceous chondrites. And carbonaceous chondrites have come from space and it turns out contain amino acids. Now, if you think like I think, you probably think to yourself, well, wait a second. If these fell out of the sky, maybe on the way down it like hit a bird, right? And those amino acids that are in there are just from that bird that the meteorite smashed into. It has nothing to do with being amino acids from space. We're pretty sure, though, that those amino acids actually were abiotically formed in space. Because, if you remember, amino acids are chiral. They come in a right-hand and a left-hand form. Living organisms, well, we only use one of those forms. The amino acids or carbonaceous chondrites contain both forms. And so, it seems like it's a possibility that maybe we don't have to worry about how amino acids would have developed abiotically here on Earth, but maybe they were brought in here from outer space. Maybe that was the origin of those. And so the question of Stanley Miller's experiments is a moot point, because maybe they didn't come from here. All right, so whatever way we got them, either in the early atmosphere they developed, or perhaps brought in by carbonaceous chondrites hitting the Earth. Just having an amino acid by itself, that's not enough for a living organism. You need to be able to put them together into polymers, into long chains that can carry out then biological functions. Most of our important biological molecules are these polymers, are a bunch of these individual units hooked together into longer chains. So there needs to be some way for that to happen in order to form these actually biologically active molecules. It has been discovered that if you take a solution of amino acids and drip them down onto a hot surface, that they will actually start to form bonds among them so that they could connect them together and have a large molecule. This is especially true, it turns out, if you're dripping them down onto clay for some reason. Now, while this seems to be the case, I will mention that the molecules that you get if you drip a solution of amino acids onto clay are not going to be exactly like our proteins. That there's a lot of cross-linking that takes place, and it's not just those standard peptide bonds that develop. But it at least shows that it's possible to make those connections, even if they're not the same exact sort of connections that our current living organisms use. Okay, so let's imagine that we've gotten to the point of forming macromolecules, however that happened. Now we need to put them somewhere, specifically in what we're going to call a protocell, which is basically a membrane around those macromolecules. This is going to divide things into an inside part and an outside part. Now let me mention why this is so important. Why do you need to have an inside versus an outside to have an actual living organism? It allows that organism to maintain a different internal environment than the outside world. So I'm thinking here specifically of something like pH, for example. The pH of a solution has a big effect on what sort of chemical reactions take place. And living cells tend to maintain their pHs usually around pH of 7-ish. In order to do that, you need to have an area that you can separate out from the rest of the world. And so having a membrane creates this separation between the internal environment and the external environment. I mean, you know how it is. You don't want to have your insides outside, and you don't want the outside world in your insides. Same sort of thing here. Well, for this one, it turns out that if you take a handful of a molecule called a phospholipid, take a whole bunch of phospholipids and throw them into water, they arrange themselves into a bilayer, into this two-layer membrane, which will actually end up being a circular membrane just because of the chemical interactions with water. And so if phospholipids are around, you can expect a bilayer to develop, resulting in this container that could go around a macromolecule. That macromolecule then can start carrying out chemical reactions, right? You could have a metabolism. All right, not quite enough. We need one more step here. We need to have self-replicating molecules. Molecules that can make copies of themselves, and then so that information can be passed on from parents to offspring. Well, it's been discovered that RNA can function as an enzyme. We're going to call that, when that happens, a ribozyme. So most enzymes are going to be proteins, but not all of them. Ribozymes are RNA enzymes. Now, it also turns out that one of the functions of a ribozyme is that some ribozymes can actually make copies of RNA. This may have allowed self-replication, right? In other words, may have allowed a molecule to copy itself, so there are multiple copies of it. Now, I will mention that this sort of thing has been accomplished, pretty much accomplished in a laboratory. Not exactly. What they've done in a laboratory is they've got a couple of ribozymes. One ribozyme can copy the other one, and the other one can copy the first one. And so they can end up copying each other. It's not one copying itself necessarily. But it's not too far to imagine that process completing where one can just copy itself. Once that starts happening, once you have self-replication, then you can basically have natural selection, right? Because some of those ribozymes will be better at copying itself, some will be faster and more efficient, require less energy. That means that there can be selection amongst those different ribozymes, resulting in all these differences, these changes, these mutations can then take place. And so you can have a more efficient method of transmitting information from parent to offspring as those ribozymes copy themselves. Now, this has led to an idea known as the RNA world hypothesis, which basically says that the earliest organisms probably used RNA as their genetic material. And then at some point, there's this switch over to the world becoming DNA dominated. Right? And so starting out with the RNA world hypothesis, and then eventually DNA becomes the genetic material used by all living organisms. So I have a short video here that is going to show that person who's doing research in that and what she's done.
It still fascinates me.
The incredible beauty of the universe.
I call it a marathon and not a sprint.
You never wanted to be in second place.
Whatever I wanted to do, I felt that I could do it. I like what I do. I am so grateful that I've been able to pursue my passion for 40 years and get paid to do it.
For 4 decades, Joanne Stubbe has relentlessly pursued fundamental biological processes, but with a chemical point of view.
My grandfather used to take me out in the woods. I knew where all the wildflowers and birds were. I earned my money by selling crawfish and hellgrammite bait, so I was excited about biology.
Scientists believe that long ago, RNA molecules were the dominant life force. But how did this RNA world transform into today's double helix world? Stubbe theorized that an enzyme, ribonucleotide reductase, must be the link, converting the building blocks for RNA molecules into the building blocks to make DNA.
I've always been fascinated by enzymes. They're amazing, and chemists can't come close to recreating these kinds of catalysts. Right now, the holy grail for me is trying to understand how ribonucleotide reductases and bleomycin all use metals involved in catalytic transformation.
There are biosynthetic pathways that have specifically evolved to incorporate metals into enzymes and natural products. Stubbe believes that learning how these metals are incorporated will provide new targets for design of anti-tumor agents. RNR is a proven target for a number of diseases.
Before I retire, that's what I really want to do, is understand how the metal clusters are assembled. And so that's what's driven me all these years, is just dying to know the next answer and solve problems.
So one thing I just want to point out, just when you think about how complicated this class is, just don't forget that we're not learning this sort of ridiculous physiological pathway that we see in this video.